Hello, judges. I'm Arshika Yalavarthi. I'm a senior at Barrington Community High School. The following research studies the various potential drug and natural inhibitors of neurodegenerative pathways in amyotrophic lateral sclerosis. Over the course of this project, many individuals have helped me overcome challenges and find ways to derive conclusive results. I would like to thank my mentors, including Mrs. Foley, Mr. Beck, Mrs. Turngren, Rishi Ray, Ella Holterman, and my family for supporting me in this quest to investigate ALS. The symptoms of ALS on patients are cognitively and physically debilitating. As there is little known about this disease, the purpose of this project is to shed light onto the potential treatment plans. This research aims to investigate the potency of known drugs that are currently being used in the treatment of ALS and its symptoms in inhibiting specific proteins. To explore if drugs with similar chemical makeup can inhibit the proteins causing ALS degeneration, and to prognosticate whether natural compounds that have been used to treat ALS have significant backing in preventing enzymatic reactions. The three hypotheses for this research are the following. If the known drugs can bind to the active sites in the receptors or proteins, they may be able to inhibit the receptors. If the other chosen drugs have similar structures as the known drugs, they are more likely to inhibit the mutated proteins in ALS. If the natural compounds have an abundance of proteins inhibiting compounds, it is possible that they can be used to treat ALS. ALS's symptoms include muscle spasticity and weakening, loss of function in limbs, and progresses to paralysis or respiratory failure in the later stages. This is predominantly caused by the apoptosis or death of lower and upper neurons. Upper motor neurons, starting at the cerebral cortex or brainstem, convey messages on movement to the spinal cord, while lower motor neurons convey this information from the spinal cord to skeletal muscles. The damage to motor neurons causes muscles to atrophy, furthering the physical deterioration. ALS can be passed down generations through autosomal recessive, autosomal dominant, and X-linked inheritance patterns, or occur sporadically due to environmental factors. The hereditary mutations in genes encoding proteins or enzymes causes degenerative pathways. The aggregation of mutated proteins in neurons inhibits the neuron's functions, leading to the symptoms of ALS. The enzymes studied are superoxide dismutase, transactive response DNA binding protein 43kDa, and fused in sarcoma that form inclusion bodies of protein aggregates. SOD1 is a homodimer of metal, copper, and zinc cations. It primarily reprocesses harmful superoxide byproducts or free radicals from mitochondrial oxidative phosphorylation, creating hydrogen peroxide. Mutations in SOD1 invoke mechanisms such as glutamate exotoxicity, disruptions of axonal transport and mitochondrial function, and reversible inhibition of other proteins within aggregates. TDP-43 and FUS both regulate transcription of DNA into RNA and stabilize mRNA before translating it into proteins to carry out bodily functions. Mutations in these proteins causes modifications in how mRNA is translated into proteins, mutating other proteins. FUS specifically also transports mRNA from the nucleus for transcription into proteins and repairs DNA. Mutations in FUS thus also leads to impaired cell communication, protein production and transport, and DNA correction. Molecular inhibition is a way to study how these enzymes are deactivated and no longer able to cause further harm to the body. In this process, researchers introduce and bind inhibitors to active sites of the mutated enzyme where the enzymes are to carry out their tasks. This binding, often through non-competitive and reversible inhibition, stops the enzyme from mutating other molecules and deteriorating the body by hydrogen bonding and preventing the other molecules from even interacting with the mutated enzyme. However, these inhibitors are not always accurate in their binding, as they may bind to perfectly functioning enzymes that are similar to the target. The possible inhibitors tested were two approved drugs and inhibitors, edaravone and riluzole, two similar drugs to the known ones, DMP and AMB, and a traditional natural remedy treatments, creatine and gallic acid in poo or tea extract. Edaravone removes free radicals in the body and neutralizes a chemical imbalance as an antioxidant to alleviate oxidative stress and cell apoptosis. Riluzole blocks the excess glutamate excitatory neurotransmitter release to prevent overstimulation of the brain and degeneration of motor neurons. Creatine promotes adenosine triphosphate reprocessing in muscles to provide energy to the body and has biosynthetic pathways that may cause better motor efficiency and extended survival in ALS patients, while alleviating glutamate excitotoxicity. Pu'er tea extract impedes the advancement of cancer FET proteins and degrade FUS proteins, inhibiting protein aggregation. Gallic acid in pu'er tea extract forms many hydrogen bonds with other molecules, acting as a neuronal protector. To test some possible inhibitors and their effectiveness in binding to active sites of the enzymes, docking software can be efficient. When drug testing, the timely, precise, and cost-effective manner allows researchers to isolate molecules and study their interactions through binding energies and number of hydrogen bonds. The independent variables in this project were the different receptor-ligand pairs, while the dependent variables were the binding energy and the number of hydrogen bonds. The constant variables were the software environment and procedure, and the control or baseline trials were the ones with the known inhibitors, or drugs. The materials were a computer, web browser, UCSF Chimera, and Autodoc Vena. It is also recommended that you use an ocular lubricant drops as a safety precaution due to this screen and blue light exposure. The procedure was to dock each of the 3 enzymes with each of the 6 potential inhibitors on a molecular docking software called Chimera. The interactions from these dockings were studied to record how the binding energies or scores and the number of hydrogen bonds compared to the control trials. Each enzyme and ligand pair were docked 3 times and the results were averaged. The full procedure and manual for Chimera is listed in the paper. As computer-generated data was used to compute these results, the predicted data may be erroneous in comparison with lab testing. The error was minimized by the repetition of each of the combinations of enzyme and ligand pairs 3 times. Comparable or more negative binding energies and comparable or more number of hydrogen bonds signify that the compound is likely to be an inhibitor as well. When the error bars overlap, the data's difference is statistically insignificant and the properties are comparable. The combinations were classified high confidence if they had more negative energies and more hydrogen bonds. If only one condition was met, they were called medium confidence inhibitors. If none were met, they were low confidence inhibitors. The data is as follows. In the case of SOD1, DMP and gallic acid had comparable or lower binding energies than the control, but AMB and creatine had high energy. In addition, DMP and AMB and creatine and gallic acid all had comparable or higher number of hydrogen bonds with SOD1. For both TDP-43 and FUS, only creatine had higher bonding energy and all had comparable or higher number of hydrogen bonds with TDP-43 and FUS. Overall, DMP, AMB, and gallic acid should be further tested to explore its effects on amyotrophic lateral sclerosis patients. However, creatine should be studied on computers before being tested on patients. As predicted, ligands that bind at specific active sites, drugs with similar structures as the known inhibitors, and natural compounds with protein-inhibiting compounds were more likely to be inhibitors of ALS enzymes. Thank you for your time.